Okay, so let's pick up where we left off. And what we saw here was fructose 1,6-bisphosphate being converted into fructose 6-phosphate by the fructose 1,6-bisphosphatase enzyme. Now this enzyme works primarily on substrate availability. So as the concentration of fructose 1,6-bisphosphate increases, because we are again feeding the gluconeogenic pathway, this enzyme will become active, converting that fructose 1,6-bisphosphate into fructose 6-phosphate. So now, first level of regulation of PFK1 so that we can prevent this reaction, prevent that futile loop, is the negative regulation by ATP levels. Okay, so again, this is another counterintuitive step. We are in a fasting state in liver, so you would think that energy levels are depleted. That would be the case primarily in skeletal muscle or other cells, but in the fasting liver, remember, we have beta oxidation of fatty acids generating lots of acetyl-CoA, so our ATP levels, our energy state in the liver, is extremely high in the fasting state. So this beta oxidation is going to keep ATP levels, concentrations, very high, and this will inhibit PFK1 activity, preventing that futile loop. The other aspect that controls PFK1, because this is not sufficient for this, because we need to make sure that gluconeogenesis fires in one direction, is going to be the conversion of fructose 2,6-bisphosphate into fructose 6-phosphate. So remember that we have PFK2 activity involved in here. So what we have seen before is fructose 6-phosphate being converted into fructose 2,6-bisphosphate, which again, remember, is not a glycolytic intermediate, but this is involved in the positive regulation of PFK1. So what is going to convert this here is that we are going to have PFK2 in its active state to convert this, and this happens in glycolysis. Now, in gluconeogenesis, we need to reduce the concentration of fructose 2,6-bisphosphate as an activator of PFK1, and so in order to do that, we need to reverse this reaction to cleave the phosphate off of the number 2 carbon of fructose 2,6-bisphosphate, reducing its concentration and reducing the activation of PFK1. This is also going to be done by PFK2 because this is considered a bifunctional enzyme. Being a bifunctional enzyme, it has both kinase and phosphatase activity, fructose bisphosphatase 2 activity. So let me include this portion of the activity on this side of the equation. And this, in glycolysis, is inactive, so that the kinase activity is active, and we are converting fructose 6-phosphate into fructose 2,6-bisphosphate using ATP as the substrate for the high energy bond. Now in the reverse reaction in gluconeogenesis, the PFK2, the kinase activity, is inactive, and the phosphatase activity is active, and this is releasing inorganic phosphate. So now the question is what converts the activity between these two bifunctionalities in this single enzyme? Well, remember, we are now in a fasting state. So you've already guessed that glucagon signaling is going to alter the activity of this bifunctional enzyme. So with glucagon signaling, that will activate PKA, protein kinase A, and that is going to phosphorylate the enzyme. And by phosphorylating the enzyme, let me put this up here to indicate that the whole enzyme is being phosphorylated, that is going to turn on the fructose bisphosphatase activity and inactivate the kinase activity. So now that we can convert fructose 2,6-bisphosphate to fructose 6-phosphate, reducing the activator's concentration in gluconeogenesis. The other direction, in order glycolysis, this is going to be insulin signaling. So this is insulin going to drive the activation of a phosphatase enzyme. This phosphatase now will remove the phosphate from the PFK2 bifunctional enzyme complex, which will activate the kinase function and inactivate the phosphatase function, allowing the normal reaction to occur in PFK2 to increase the activity of PFK1. Okay, now, so we've got our PFK1 inhibited, so we're generating our fructose 6-phosphate. You know the next step, the phosphoglucose isomerase, making glucose 6-phosphate. And now we have to take this to glucose in order to complete gluconeogenesis. Okay, so we have another problem. This reaction, we normally use a hexokinase, glucokinase in the liver, and this reaction uses ATP to form glucose to glucose 6-phosphate, so this reverse reaction is irreversible. So what we need is another enzyme to take glucose 6-phosphate and convert it into glucose. And the enzyme is called glucose 6-phosphatase. Now this enzyme is going to work primarily on substrate concentrations. So again, same principle as we've seen before, the glucose 6-phosphate concentration is going to increase because we are feeding the gluconeogenic pathway from below, and so that's going to make a rather high concentration of glucose 6-phosphate, and the glucose, once this converts, is going to exit the liver cell through GLUT2, because GLUT2 is the transporter in the liver, and it works based on high glucose concentrations, which they'll be relatively high because we are in the process of gluconeogenesis. This will put glucose in the blood, which is at a lower concentration, because we are in a fasting state, and so we have a gradient that will drive the reverse reaction of glucose transport into the blood. Now, why won't glucokinase work in this case? Well, again, recall the activity of glucokinase. So if this is glucokinase activity, remember the Km of this enzyme. The Km is out here. So if glucose concentrations are low, the activity of the enzyme is going to be low. And this is exactly the case in gluconeogenesis, because glucose is being transported out rapidly of the cell through GLUT2, and that is keeping the glucose concentrations low in the intracellular compartment of the liver. And so glucose kinase activity is low under these cases. Contrast that with high concentrations of glucose 6-phosphate, which is going to increase the activity of the phosphatase enzyme. So it's going to promote this reaction into the phosphatase direction. Glucose leaves, keeps glucose kinase activity low because of low substrate availability. What we prevent, again, is the futile loop. Because if the glucose wasn't transported out of the cell, its concentration would increase. Glucokinase would become active. We would form glucose 6-phosphate, which would now be dephosphorylated by the phosphatase enzyme, and then we have that futile loop. So based on the high Km of glucokinase, this is going to prevent this futile loop because the glucose is rapidly leaving the liver cell and into the blood. Now there's one more piece of information. Oh yes, there's always one more piece of information. And I've got that here somewhere. Where are we? Oh yeah, there it is. You can imagine my desk is a mess trying to explain this. Okay, so now, what are the gluconeogenic precursors? In other words, what carbon sources are being used by the liver in order to generate glucose? So the liver has to get these carbon sources from somewhere. It can't continually deplete its own carbon sources to provide glucose, or the liver will go into malfunction. So what we saw at the fork in the road is pyruvate. So pyruvate is going to, remember, in gluconeogenesis, form oxaloacetate by the pyruvate carboxylase enzyme. So the question is, and I started with pyruvate, but where does the pyruvate come from? Well, the pyruvate's going to come from extracellular sources. So there are three major gluconeogenic precursors. The first one is lactate. So lactate remember, is a byproduct of red blood cell metabolism, because red blood cells are unable to perform oxidative phosphorylation because there's no mitochondria, so they convert pyruvate into lactate in order to regenerate their NAD, and in the process the lactate is secreted out into the blood. The liver picks this lactate up and converts it into pyruvate by the lactate dehydrogenase enzyme. So this lactate dehydrogenase enzyme is the same enzyme that we find in muscle under anaerobic conditions, the same enzyme we find in red blood cells that allows it to regenerate its NAD capacity. And in this case, in the liver, we're going to take this lactate and convert it, use the carbon sources for pyruvate. And in the meantime, we'll take a couple little electrons in there for energy. So lactate is the first source. Another source is alanine, the amino acid alanine. So if we look at the structure of alanine, we can see here that there is the potential for using a carbon backbone. But alanine, we have to take one step and process this nitrogen. And the processing this nitrogen is going to be done by what's called a transamination reaction. So we are essentially going to remove that nitrogen and replace it with a ketone. And in doing so, we generate pyruvate. We'll talk more about transamination reactions when we get into amino acid metabolism, because this is going to be intimately connected with the urea cycle to get rid of the nitrogen. So lactate and alanine are two major contributors to pyruvate that will provide carbon sources for gluconeogenesis. The other one that we have to concern ourselves with is to pick up on adipose activity in the fasting state. Remember this molecule. This is called glycerol. Glycerol is released by adipose in the fasting state because of triglyceride hydrolysis. Remember, triglyceride hydrolysis releases 3 fatty acids and resulting in the glycerol backbone. The glycerol backbone is then put into the blood. The liver picks this up, and then the liver is able to make glycerol 3-phosphate. This process is done by the enzyme glycerol kinase. And this is an enzyme that is rather unique to the liver. And it's going to take glycerol, and it is going to add a phosphate group, hydrolyzing ATP, to form glycerol 3-phosphate. This process, you'll see in textbooks, is also called gluconeogenesis. We'll see this process again later on in the course when we talk about lipid transport. But the glycerol 3-phosphate in the liver, remember the glycerol 3-phosphate dehydrogenase enzyme can convert this into DHAP by a simple redox reaction. And generate a little bit more NADH for the liver in a fasting state. The DHAP will then isomerize into glyceraldehyde 3-phosphate, and then these two combine into fructose 1,6 bisphosphate and on up we go to glucose synthesis. So this is where glycerol, coming from adipose in the fasting state, is reutilized from the backbone of a triglyceride in adipose, and the liver picks it up so as not to waste carbon units and contributes that into the gluconeogenic pathway. Okay, so that is gluconeogenesis, the reverse reactions of glycolysis, and the 3 reactions that have to be bypassed in order to prevent that futile loop, and also to prevent, to get around those irreversible reactions. So this is going to take a little bit of study time, and I would encourage everybody to draw these maps out in their entirety in your own hand, and we'll go over this in more detail in class to cover some of the main concepts, especially the regulation points. Those will be the most difficult.